Good afternoon to everybody. This is the May session of the young seminar organized by the Italian Society of Statistical Physics. And as usual, we have two speakers. Today we have Federica Ferretti and Claudia Nicoletti. The first speaker will be Federica Ferretti, and just a few words to present her and the talk. Federica is affiliated to the Simons Institute for Theory and Mathematics in Biology in Chicago. And she got a master and PhD thesis at Sapienza University with the supervision of Irene Giardina on clocking dynamics in collaboration with Alexander Balzak and Thierry Mora of the École Normale Supérieure in Paris. And since 2022, starting in 2022, she got a postdoc at MIT on statistical models for immunology. And since last year, she's at the Simon Institute in Chicago and working on statistical modeling of biological learning processes. And today, Federica is going to give a talk with the title Implicit Regularization from Single Cell Variability in Evolution. 25 minutes, Federica, please. No, il tuo microfono non è proprio silenzioso. Sembra proprio silenzio. Oh,
yeah,
okay.
Sorry. Okay, sorry, my bad. All right. Okay, in case you cannot see the screen, just let me know. So thank you very much for the kind introduction, and of course, thanks for the invitation. I'm very pleased to have the chance to present this work. So I have decided today to speak about something which I'm trying to wrap up right now. It's not finished or published yet, and it's something I have been thinking about with Arvind Murugan from University of Chicago and Miran Kardar from MIT. And the spirit of the work, as you'll see, is just to think and try to bring some concepts which are quite established in the context of machine learning and especially deep learning to the realm of evolutionary biology. So to start, I want to try and convince you that evolution can be thought of as a learning problem. And the reason for that is that evolution is based on the principle of the survival of the fittest, and this is nothing but an optimization principle. Darwin's theory of natural selection tells you that organisms with traits that are beneficial in a given environment they have higher chances of surviving and reproducing, and as a consequence, these traits that are beneficial will increase their frequency in the population over time. So this is not just a theory. It was over the centuries, especially after the introduction of sequencing techniques, it's been proven, validated in the lab. And it allows you to think of evolution as, especially in certain regimes, as a stochastic trajectory in some fitness landscape that tends to climb uphill in this landscape. Now, to make the analogy complete, you need to notice that being fit in an environment is not the end of the story for biological systems. It is also important that they are robust against environmental perturbations. And so this problem is analogous to the overfitting or generalization problem in learning. And the reason why you want to be robust is that if the population has evolved very well in the sense that it's very sharply localized around the peaks of the fitness landscape, if the environmental perturbation washes away the peak, then the population is in a bad spot because it's unfit for the new environment, and this can lead to extinctions. So one example in biology where this is particularly relevant is the adaptive immune system. You know that mammals have both an innate and an adaptive immune system. And the goal of the adaptive immune system is precisely to learn about pathogens that an organism encounters throughout its lifetime. You can think of these landscapes as sort of binding landscapes, which express the binding affinity of the antibodies that you produce towards some pathogen. So this learning about the pathogens that you encounter happens by means of an evolutionary process that occurs in the lymph nodes, and at the end of the process you produce antibodies and memory B cells. So there is a selection for those that bind the best, but viruses or pathogens in general can mutate. And so if the population of antibodies that you produce is has a very narrow specificity for the first virus that you encountered, you might not be protected against mutants, and you may need to produce new antibodies. So there's no point in having built this immune response in the first place. So what you want to do is to skew the immune response towards these lower peaks that are conserved across different mutants, which correspond to the generalized antibodies. So this is just a motivation and very cartoonish explanation of why we might be interested in these problems. And I wanted just to point out that there is the same tension at play in biology and in learning problems between optimization and generalization or avoidance of overfitting. Now, in the context of machine learning, people have found strategies to cope with this problem. And these are regularization strategies, which can be classified into two main categories. Explicit regularization, if you add an explicit regularization term to the loss function, or implicit regularization, if the strategy consists in acting on any of the other elements that appear in this equation. And so this could be transformations of the input data, choices of the model that have to do with the choices about the architecture or choices about how you define this error function, and strategies that act on the optimization procedure itself. And I want to draw your attention to something specific in this class, which is stochastic gradient descent. Stochastic gradient descent has attracted significant interest, especially in the early 2020s in the statistical physics community. And it consists in a simple modification of gradient descent, of standard gradient descent. So instead of computing the gradient of the true loss, the full loss, which is just an average over contributions that come from each point in the training data set, you approximate this loss with an approximate loss that is the average over a subsample of the training data set, the batch. And this batch is stochastically sampled at each learning epoch. So this introduces a little jiggle, a little stochasticity, which is, it turns out, is beneficial, at least in certain regimes, to improve the generalization performance of the network. And this improvement is especially evident when the size of the batch is small compared to the total size of the training data set. So this motivated the introduction of stochastic mini batching procedures, which are now used basically in every application. And as I was saying, the involvement of the statistical physicists helped in understanding why this might be the case, why this stochastic effect can be beneficial to improve generalization. And the reason, in a nutshell, is that this mini batching creates a multiplicative noise that biases the solution towards flatter regions of the loss, creating this inverse variance flatness relation, which is the opposite to what you would expect based on equilibrium thermal arguments. So the question we had was whether similar strategies could be a play in biology. And to do that, what you need to do is to move from the picture of a single fitness landscape, a fixed fitness landscape, to an ensemble of fitness landscapes. And what we're modeling here is nothing but single cell variability, because what we're saying is that if you have a population, an isoclonal population, meaning all the individuals are identical from a genotypic perspective, you don't have a single fitness value that is associated to this genotype, but a distribution of fitness values. And each individual can sample from this distribution. So it isn't hard at all to convince biologists that this is a good ingredient to incorporate in your model, because single cell variability is very ubiquitous. But in this context, I want to just mention 3 scenarios that motivate this choice. The first scenario is in the lab. So you can imagine of imposing by hand these environmental heterogeneities such that each cell at each generation experiences a slightly different landscape. The second scenario is when you're interested in building these genotype-to-fitness maps from quantitative data, like deep mutational scans, which, however, basically aim at reconstructing this part of the map. So they measure phenotypes of the proteins, properties of the proteins that are encoded by certain genes. But what you sequence is just the gene, or more often a fraction of the gene, and some property like binding affinities. If you want this to be a proxy of the fitness of the cell, so in other words, if you want to consider the role of the protein in the context of the cell, then what you're doing by considering this a proxy for fitness is actually marginalizing over a lot of latent variables that describe the state of the cell, of all the other things in the cell that can interact with your protein of interest. And so this creates naturally some stochasticity. Finally, the last example is that of affinity maturation, which is the process through which you produce antibodies. I'm not going into the details of the process, but I just want to mention that there is a complicated selection step which occurs by a sequence of reactions. And so you don't have a single number, single rate that controls the process, but you naturally have some stochasticity in the rates if you think of them as capturing a more detailed sequence of reactions. So this is just to motivate the choice of considering ensembles of fitness landscapes. And we were interested in understanding the effect on evolutionary dynamics. So we picked a paradigmatic model of evolutionary dynamics, which is the Wright-Fisher model, which is a model that describes evolution of populations with fixed size. And it is just a Markov chain where the probability of going from one generation, from a state of the population in one generation to the next generation is described by multinomial sampling. And the probability of success, the probability of success of each species in the population depends on the abundance of the species in the population and on their fitness. The tweak that we add is to consider not a single value of fitness for each of the genotypic species, which are indicated by these letters here, but a distribution of fitness values. And so for each individual, we now need to sample from these distributions. And this is the first step, which is analogous to a mini batching step. It's a sampling step. Then once we have these fitness values, we can apply the Wright-Fisher selection step, which models replication and selection. And after that, we reset the fitness values. The reason is that we want the next sampling to be independent from the previous one. You can also relax this assumption, but it is easier to work with independent samplings. And then if one wants, one can also add genetic mutations on top of that. So at the end of the day, the process is a composite Markov process where the transition probability is described as the convolution of these transition probabilities associated to the substeps. Now, this is nasty looking, but we can make some analytical progress by working in the diffusion approximation, where, which is roughly valid when the population size is large and a few other assumptions are satisfied. In this approximation, the dynamics is described by a stochastic differential equation, and it allows us to characterize the effect of this extra source of stochasticity that comes from individual mini batching. So as you can observe, the correction terms are in blue here, and we have both a correction for the drift and a correction for the diffusion term. And both these corrections depend on the variances of the distributions of the fitness values. What I want to remark is that these corrections are of order 1/n compared to the leading order term of the drift in the reference process. The reference process is the evolution of the population in the mean fitness landscape, a fixed mean fitness landscape. So you have a correction for the drift that is already of order 1/n if you compare to the reference drift, and a correction to the diffusion, which is of order 1 with respect to the reference diffusion, but this reference diffusion is the demographic noise, so it is already of order 1/n compared to the drift. And so the conclusion seems to be that effects are of order 1/n, and so they are negligible for large population sizes. However, what we observe in the simulations, then the results here depend on the specific properties of the landscape, but I'm using this example to explain the more general mechanisms. And the observation here is that there are intermediate values of population sizes where the behavior is deviating a little bit from the trend 1/n that you would expect. And this is something that is particularly relevant for certain observables, which are like several orders of magnitude, have an effect in relative change that is several orders of magnitude larger than what we would have expected. So this is true not just at the level of mean values, average values at steady state, but if you look at the distributions, you may observe that there are some shifting of the modes or emergence of secondary modes in the distribution for these intermediate population sizes. So what is the mechanism that leads to the creation of these secondary modes? And the mechanism is basically a non-equilibrium trap. You can go have a look at the equations again, equations of motion. And it is true that the drift is what dominates the dynamics, but once the system reaches steady state, we may think of the system as being localized on states which have a low value of the drift. So if there is a manifold or like a collection of points where these drift, these deterministic forces are not too big, so of order say 1/n, then they can compete with the diffusion, so with the stochastic forces. And the fact that the diffusion is non-homogeneous can create, can skew the distributions within this manifold of near equilibrium points. So this is precisely what we observe in this example. And it is easiest to look at the one-dimensional projection of this stochastic differential equation onto the first principal component of the states that are visited at steady state. So this is what you have on this right-hand side of the slide. And on the vertical axis, you have the deterministic drift, and the color code represents the temperature, the amplitude of the fluctuations projected on this first principal component. And as you can see, we have roughly a fixed point, a stable fixed point, where the drift intersects 0 with negative slope, and a marginally stable fixed point where the temperature is now much lower. And so the fluctuations here next in the vicinity of what would be deterministically the stable fixed point are stronger and able to make the system rattle away from this point. But when it reaches this other point, the temperature is much lower, and so it can stay there. So this is just a simple thermophoretic argument, which is of the same kind that motivates the effects observed in stochastic gradient descent, and can explain the origin of these secondary modes. So To summarize, I think I've been very fast. Sorry about that. We have a simple toy model where we can incorporate single cell variability in evolution, and we argue that this can be seen as a form of implicit regularization. There are few necessary ingredients to observe effects that deviate from the amplitude 1 over n that one expects, which are that the population size has to be mesoscopic, so large enough to let the diffusion approximation be valid, but not too large, because there's always a size over which you can let the effects be vanishing. The fitness landscape has to be complex. Of course, the larger the complexity of the fitness landscape, the more relaxed can be the assumption on the population size. And there must be heterogeneous fitness distributions, because this is what creates these gradients in the temperature that we are exploiting to create the effect. There is a caveat, which is that which of these marginally stable or metastable states you're selecting by changing the stability of these states by playing with the temperature depends on the constitutive mean variance relation of the fitness distributions of the system. And this is something that you need to either try to measure from the data or to analyze from toy models that model what is the source of single cell variability in the system. And so one skepticism that one can have after this caveat is that this is maybe something that is too artificial, that doesn't really happen in nature. But to this point, I want to mention an experiment that was done in the lab of Oskar Alacek at Berkeley. And in this experiment, they considered colonies of bacteria, different mutants of bacteria, where they knocked out different genes, and the knockout of the genes caused the bacterial colonies to grow differently. But they also observed, in addition to this difference in the growth rates, a variability in the demographic noise. And this was the end of their paper, like the end of the observation. But in addition to the fact that there was a positive association with the growth rate, so with colony traits, but not with single cell traits. And I think our model explains that there could be an explanation for this observation of different demographic noise amplitudes based on the fact that the knockout of the gene can also change the level of noise in the cells, the level of single cell variability, in other words. Finally, I want to mention some possible applications. There are obvious applications in synthetic contexts, like genetic algorithms, which are not used honestly to train networks, but they are increasingly used to optimize over the hyperparameters of neural networks. And they can also be used, not quantitatively maybe, but at least qualitatively, to inspire evolution design. For example, design of immunogen cocktails for vaccines, which are now just using few different strains. For instance, in the case of the vaccine, the booster for COVID or the influenza vaccines, you just have 2 or 3 strains, but maybe like moving to a regime where you really have a cocktail of many strains can be a regime where this effect of noise can be leveraged. So with this, I conclude. I want to thank the people I interacted with over the past year, especially Meran Kardar and Arvind Murugan for this project, the Murugan group and the Ranganathan group U Chicago, Arup Chakraborty and his group at MIT, who taught me a lot about biology, and the NITMB fellows at the National Institute for Theory and Mathematics in Biology in Chicago. And thank you all very much for the attention.
Thank you very much, Federica, to you. Okay. Are there questions, comments?
I have one question. Should I raise my hand or?
Which is not necessary. You can just speak.
Yes. Okay, so you mentioned, Federica, the regimes where your approach can be useful. Okay, so intermediate sizes, and this was very clearly displayed by the graph that you showed. And the other criteria related to the heterogeneity of the fitness landscape, if you want. Okay, so I was wondering, can you quantify a little bit better these regions? I mean, can you give some arguments to say, okay, if your system is like this, I don't know, some observable which lies within some range, then we can use this method, otherwise we can?
It is quite hard. So I haven't found anything that is sort of clean, because the details can matter, as I was saying. So I can write down formally what are the conditions. I haven't managed yet to find like a useful formula, if you want, or condition that can be related only to collective observables or macroscopic observables that can be defined at the level of the population without knowing some details of the landscape.
Okay.
So it has to, I think my understanding so far is that you need to have these extended regions where the amplitude of the barriers are bounded. And in the absence of demographic noise, the barriers, in the absence of corrections to the demographic noise, the barriers are most like a leading order. They are what determine where the system is going to be. But once you turn on the individual mini batching, this creates stronger inhomogeneities in the diffusion, and this can bias the points that you're going to be. But yes, it is a complex interplay between the things, and I haven't been able so far to characterize it more cleanly.
No, but I mean, if people gave you the data, okay, so it would be nice.
Of the landscape, yes, but then this would require really having proxy for what is the variability, the single cell variability for each genotype. So I think I could give you a prediction of how these bacteria that were grown in different colonies. So you have one species per colony in the experiment that I showed at the end. If you put them in competition with each other, which one is going to win? So this is the kind of prediction I can give you.
Okay, thanks.
Okay, other questions or comments? I have a very small one. In the introduction, when you introduced the stochastic gradient descent, you told that the goodness of this method is related to the generation of multiplicative noise.
Yes.
Can you say something more?
So
just to clarify.
I'm not very expert on that, but so there have been works showing that this effect of stochastic mini batching can be rewritten here in the limit of small learning rates. So this equation can be transformed into a stochastic differential equation with a multiplicative noise, where the coefficient depends on the concavity of the loss. And so this is what biases the solutions to go to these flatter regions. I'm sorry, I don't know if this answers the question.
Okay, I know that is a technical question. Okay, if it seems there are no other questions, comments, so thank you, Federica. Thank you again.